All right, we're going to get started now. Just while everyone is finally settling in, I see the numbers still ticking up in attendance and getting their coffee going. I want to just spend a second to go meta on you. It's been a long and busy journey to get to this point, and there's nothing like the motivation of walking into your office in the morning and going to your home page and seeing that ginormous countdown timer that Jessica, our marketing wizard, put up on our website. So thank you, Jessica, for that. It has been a lot of work from a lot of people to get to this point. But also thank you, Jessica, for bringing in such a big audience for this event. I know in part due to her outreach and from my blog, and also just from word of mouth, we've had a number of people already involved with this event of building this product. And to be honest, this is one of our largest, or the largest attendance we've had. We've have over 500 people register to see the launch of GenomeBrowse. To give you a context of 500 people, if we were to try to host this at a ASHG event, for example, we'd need one of those large plenary session rooms that fit a couple basketball courts, and it'd probably bust up Jessica's marketing budget pretty good to hold the wine and cheese afterwards. So thank you all for making this a big event and a big launch of GenomeBrowse. It's not every day you get to launch a new product, and I know we are all waiting, and there's a lot of speculation and rumors going around to see the launch of a new product. And of course I'm talking about GenomeBrowse, even though some of you might have been thinking iPhone 5 when I mentioned it that way. And I'm sure we'll all be attending the iPhone 5 launch later today. I know my wife is already on the rumor sites. But the nice thing about launching a software product is you don't have to wait 2 weeks for the thing to ship on over from China on a bunch of 747s. What we're going to be doing is actually sending everyone who's signed up for this an email sometime during the event of this webcast. And that email will include right in it the download links for GenomeBrowse as a thank you for signing up and being part of this launch event. That download links will take you directly to the installers. You'll get the prompted to be downloaded, and away you go. You'll be able to play with this as soon as you would like. So with that, let's go ahead and get started. I hope everyone now does have their coffee and sitting down. And what we're going to do today is talk about GenomeBrowse. We really do believe this is a revolutionary visual experience for genomic data. My name is Gabe Rudy. I'm the Vice President of Product Development here at Golden Helix. And to get through just a quick logistics of being in a webinar, you can see on the right it's very easy for you to type in messages. Please feel free to use the questions box, and as you're going along and seeing things, queue up those questions. We'll spend hopefully 5 or 10 minutes here at the end. I'll try to time my whole presentation and demo so that we have a little bit of time to answer those questions. If your questions don't get answered, and I might not be able to get through all of them, feel free to email us at our general email addresses, or me personally at rudy@goldenhelix.com. We will make this webinar available as a recording soon after. Our marketing wizard is very good at doing that, and you'd be surprised at the turnaround of that. But we will record this and make it available to download and send you out either an email, or you should be able to find it quite easily on our website. So, with that in mind, this is what I'm going to cover today. I am going to spend a few moments talking about who we are as a company. I do think this is important just to give you the context of where we're coming from when we talk about building a genome browser. And then, obviously, because this is not the first genome browser to be launched in the world, it is worth putting in the context of why are we building this genome browser, and what do we really hope to accomplish with it. But with that out of the way, we will spend the majority of our time actually going through using GenomeBrowse to do some real meaningful work. We're going to go through some different use cases and really spend some time showing you some real hands-on action. And through that, we really hope that you get a sense of what GenomeBrowse is all about. Okay, so who are we as a company? And I really do think this is important in the context of we have been around since 1998. That means we're not some young gun, VC funded, get in, get big, get out, here today, gone tomorrow type of company. We've really been through a lot of different technology changes, a lot of different analysis technique changes, and we have been through most of these last dozen or so years building genotype, DNA-seq, RNA-seq software, and providing analytical services to a wide range of customers. And this is actually really what I wanted to focus on, is we have a number of strategic partners, and we have a huge user base, collaborator base, stakeholder base, and this is really what we feel is probably the largest asset we have as a company. And you can't get through 12 years in this industry without really engaging and being part of that collaborative process of getting research done. And we find that being in a mode of constantly learning, accepting feedback, and using that to improve our services and our software does allow for us to really provide great analytical tools, and we are proud to be a partner with many of these both academic, commercial, and pharmaceutical companies in the genomic space. And so as part of that collaboration process, we are very grateful to be cited as a software tool and as an analytical service in a number of noteworthy scientific advances and research. And so we have to date over 650 citations in many different journals. And this is all to say that it has been a journey to be part of this industry. It's been a journey in which we've learned a lot from our greatest asset, you, the individual, the customer, the researcher, the analyst powerhouse. And that journey has involved us building and many different solutions and products, but our flagship product, which includes all of those solutions, is Golden Helix SNP and Variation Suite. We abbreviate it SVS, and it has been having workflows and solutions in this space covering things from DNA-seq all the way back to microarray and CNV analysis. And it's worth bringing up in this context as it is part of the story of how we got to GenomeBrowse. SVS has had a visualization tool, which we in fact call a genome browser, because it in fact incorporates genomic annotations and statistical results. So those results might be from an RNA-seq or DNA-seq experiment. They might be from a copy number or a GWAS experiment. But really, since 2009, we have been learning and building up the in-house expertise to both curate annotations, which we think is one of the key points here, is building up the understanding of what's important to do interpretation of genomic data, and also just to build up the engineering and support expertise to learn how to build a good visual tool that that can meet all the demands of the analytical framework. So that said, though, our tool has been based on sort of the statistical results interpretation. And when you think genome browser in the market, what you really are thinking about is getting to the meat of what is being produced on an NGS sequencing platform. And that is, you're producing millions and millions and millions of reads and aligning those to a reference genome. And those alignments are stored in a file called a BAM file, and they can be gigabytes and gigabytes large, and they can be extremely resource intensive to try to view. But this is certainly not a problem that hasn't been tackled by some extremely competent and engineeringly capable people. And I don't want to dig on other products from the perspective of, I know we all put a lot of effort into our work, and I think IGV, which is probably the most well-known and most utilized genome browser, is obviously a great engineering feat. It's been probably the default choice if you were to try to look at BAM files. And there's the Savant genome browser over here, and then there's the UCSC genome browser. And let me just go through just a couple seconds to emphasize kind of some of the things that we have seen from these tools, what we've learned from them, and sort of how people have been using them, and also the feedback we've heard of where these tools have actually been difficult to use, or where they feel like they have spending more time than they would expect. So one, with IGV, this is a great at genomic alignment, but some of the things I've heard is that it can be difficult to get your data in and get especially all of your annotations lined up. And to be honest, I still quite haven't figured out what all these different color bars on the left mean. I know they mean something, and I've tried to read their manual a couple times. But the point is this is still designed generally for a very competent high-end user. You need a high-end computer to use it. All these desktop tools are Java-based. They require an enormous amount of resources to run, but they are doing the job if you get your data in. The other component is that they generally also can be disorienting as you move around the genome. And with NGS data, and here's the really interesting thing, folks, it's not just about single nucleotide variants. I mean, the genomic context that might be useful for your analysis could be at the single base pair level. It could be at the mega base pair level. There could be features that are tens or hundreds of megabases, and that is something you don't want to get disoriented or lose, so that you might actually miss something important there. So that's something we've really noticed and want to focus on, is keeping your fluidity and orientation as you navigate around here. And the other thing that I really do need to commend UCSC for is public annotations. And UCSC is king of data curation. They have tracks for every possible use case of every possible species. They bring them in from dozens of consortiums and dozens of sources. And you can get lost in all that, but it is all there and possible to be viewed. You do have a web-based interface, so you can only have so much of an interactive experience. And the other thing is there's this compromise. Because this data is web-based, if you have a 10 gigabyte file, there's no chance that you're going to be viewing it in this format. If you have some statistical results, you might be able to put a couple pieces of data up there on a user track, but there is this compromise of being able to visualize the rich information from your NGS data and the public annotations you have from a web-based experience. So, with that in mind, and with all the things we've been hearing from our engaged customer base, as I say, is our key driver of all of our innovation, we've been trying to collect in minds what are the lofty goals of an ideal genome browser. And I do have to say, without a doubt, these are goals I can't expect with a single version of a piece of software, or potentially with any number of versions, to achieve all these goals flawlessly. But I think it says a statement about what a vision of a piece of software is, and what we would put our effort into to really help the user understand what we're trying to accomplish. And that first bullet is really key here. It's about usability, and it's about getting to that information represented in a meaningful way. If you can't zoom out to the gene or chromosome level and still have the context and the information represented to you in a meaningful way, you might lose something. You might lose something that's really useful and might actually be critical to your workflow or your analysis or your science that you're trying to move forward. So we really find that usability came up a lot as we surveyed people, and it's something we wanted to put at the very core, the very first bullet on our list. The second is, do we have to make this compromise between cloud-based annotations and all these nice up-to-date curated public data sources that can be updated on a moment's notice and streamed with the ability, with the refresh rate of a web page, and the ability to use your local samples and have the performance of a local desktop solution with that interactivity. And we think that we can break that compromise and actually achieve both of those goals. And finally, if you have to, hand over that 200-page user manual to your the person who can spend the time on there to learn something, because the learning curve is so steep, you're not getting that opportunity to get your hands on the data yourself. We think that's very, very critical, that you can, with very little learning curve, with very little investment of time, get your data in, be able to have it automatically do what it needs to get it to the point where you can investigate it and learn from it and follow up with your analyses. So that's really something we also aspire to, and it goes hand in hand with usability, but it really focuses on very few points of friction or barriers to move forward with your analysis. So with that in mind, and there's certainly any number of things you could tackle as far as use cases for a genome browser. And that's something we also are aware of. We cannot accomplish all things for all people in a first release of a piece of software, but we do think that we could focus on a couple key use cases, and we're going to actually spend our time going through these use cases today. And we will, one, cover a lot of ground, and two, that focus will provide for potentially even a better experience than if we just scattered every single feature request in there, and it tries to be all things to all people, and you start to have very esoteric use cases trying to be supported, which then again hampers that usability and ease to learn. So we are going to focus on these three use cases of RNA-seq, expression analysis, confirmation, and exploration, DNA-seq variant analysis, and interpretation. That's not to say GenomeBrowse is limited or constrained by these use cases. We have a lofty and exciting roadmap ahead of us, but this does focus on what we're going to be accomplishing with this milestone and what I'm actually going to be demonstrating today. So it's been a long journey. It's been about 12 months since we've started this. We announced at ASHG last year in a joint partnership with EA as we were building a larger RNA-seq solution that GenomeBrowse was going to be part of that solution, and that's one of the use cases with RNA-seq. And at this point, I do have to actually thank also the fact that we have been having a huge engagement and participation in an early access program for GenomeBrowse. That's been going on for a long time, but especially over the past 4 months, we've had people spending a serious commitment of time filling in our guided workflows and giving us guided feedback, and we've been learning from that guided feedback, and it really has made this a solid release, and we're very proud to be making it available to you today. So with that, let's go ahead and we will spend the rest of our time going through 3 different use cases. The first one, we will do an RNA-seq differential expression. I'm going to start from scratch here, and we're actually going to investigate a differentially expressed gene between 2 different samples and spend some time just seeing how that exploratory process works in GenomeBrowse. The second one, I'm actually going to do a variant follow-up, putative variant follow-up, and we're actually going to be doing real sample data here. Real in the sense that this is actually samples that have been graciously shared with us by Dr. Golson-Lyon so that we can reproduce his results, and in fact we were very easily able to reproduce his results. But at the read level, there's a great story to be told, and as we go in and interpret their statistical and filtering results of that analysis. And so we'll use GenomeBrowse to make that to tell that story and to validate the variants that were discovered and were in fact found to be the causal variant for a individual syndrome that Golson-Lyon's research group discovered. And finally, we're going to spend some time looking at some exome trios, and it's not just any exome trios. These are actually a single trio that I received my own exomes for. I was part of the 23andMe exome pilot project, which normally is a single sample being processed from your 23andMe, but I was lucky enough to be able to get both myself, my wife, and my son exome analysis. And if you had read my blog, you would already have seen some of the results of my analysis there. But I'm going to actually follow some workflows that follow trio analysis, and we'll follow up on the variants there, as well as touch on some of the ones I looked at in my blog. So that will be used as a platform for interpreting variants in the context of those exomes. So let's start with RNA-seq expression profiling. Just give me a second here. Okay, so what you're looking at here, and if you're familiar with RNA-seq analysis, this is not an unfamiliar graph. It's actually called a cluster graph with a dendrogram axes, and this is actually a graph produced by our software SNP and variation suite in our latest release. But what we're looking at is 20 different genes, and these are actually the genes with the highest statistical significance of differential expression across 10 different samples. As you can see, these samples clustered into the first 5 and the second 5, clearly having a nice clustering effect. A darker shade means that the gene was highly expressed, and a lighter blue means that it was low expression. And there's 2 different groups here because these 5 samples actually are one subtype of breast cancer, and these are all done using breast cancer cell lines. And these 5 samples are a different subtype of breast cancer. Now, to spend 50 seconds, if you haven't actually been that familiar with how RNA-seq works as an experiment, first I have to give great props to Expression Analysis, who's been our partner in building our RNA-seq workflows. EA actually sequenced these 10 and actually 5 more different breast cancer cell lines and provided that data as a benchmark and as a publicly available resource for people to use in their analysis. And the RNA-seq workflow starts with that sequencing, but what you're really doing is you're using sequencing as a tool to measure which samples and which genes within those samples are expressed at a high and a low level, because you're sort of sampling or randomly choosing any of the transcribed RNA in a cell. You'll see that as you looked across a genome or a transcriptome, there will be some genes that have a huge spikes of reads. That just means that that gene was expressed highly in that cell. So those reads can show up beautifully in our genome browser, and we'll show that, but those also can be turned into numbers which you can use to just test statistically which genes have a high differential expression. So in this case, for example, let's look at this GRB7. Seems to be, in this sample, 1954, it extremely highly expressed, almost to the maximum extent. Compared to all these other samples here in this subtype, it is not expressed at all, for example, in BT549. So that's what we're actually going to do, is pull up GenomeBrowse, start a new project, pull in these 2 samples, 1954 and 549, and go ahead and look at this gene, GRB7, and just see if we can understand from the numbers that were coming out of a statistical analysis what's going on in that location. Does the gene look like it's differentially expressed, and what's the genomic context around there? And that's really something you can only get from a visual experience. So I'm going to go ahead and move out of the slide mode and open up GenomeBrowse. When you open up GenomeBrowse, this will be your first experience, and I'm going to go through it just like you would. If you haven't registered before, you'll just need to register here. Remember your email and password. It will be used to access things like our community support site, which I'll mention later in this presentation. And it will be used to access things like your cloud-based content. So once you log in, which I will do right now, we will load up all of our cloud-based annotations, and bam, this is going to be your first experience just like mine here. We're looking at a brand new project. We have a couple default things shipped for you, so you have your genomic context. We're at the whole genome level of the HG19 build, looking at our cytobands along the top and our reference sequence genes. These RefSeq is the repository of extremely well annotated genes, and what we're going to do is just add in those two sample sets. We have the 1954 and the 549. Now, as I mentioned, these are publicly available. They're in this example samples tab as cloud streamable data. But I don't want to slam my internet as I'm doing a webinar, so I'm not going to load those from the cloud. But as I also mentioned, we really are focused on building cloud-based annotations that are freely available and frequently updated. We have over 105 different annotation tracks here. We can load in, for example, known gene as we go. And this known gene is going to be streamed from the cloud. It's the gene annotation that was used in this experiment. So here we go, loading it on up. And I can see the 2 BAM files are immediately loaded. And let's go ahead and move known gene to the top of my list. Okay, so, and as I zoom in here, you can see we get more and more definition. And the other thing to note is the expression profile between these samples is largely the same. As you would expect, most genes are expressed at the same level across samples, some though at a little bit higher. You see the peaks move up and down between 2 different samples. And I'm just using my mouse scroll wheel as one would feel pretty naturally inclined to do. And so here we have two samples. You can see the expression moving in. I'm going all the way down to the gene level. I can click on individual genes and see that data. Let's go ahead and search for that gene that we wanted to investigate, GRB7. There it is. I jump right to it. I hit enter. We jump to chromosome 17. You can see which region we are in the chromosome. It snaps my cytoband view, my landmark view up here to that. And let's just spend a couple seconds looking at what a gene looks like in this view. It's very important, and genes are obviously the most important part of transcriptome analysis or RNA-seq analysis because they do define kind of what the structure of the biology is in this region. So in this case, we have 4 different alternatively transcribed versions of this gene. It's the same gene, but you have different start sites. In this case, one of them starts way out here. There's another transcript that starts here, here, and here. And the thing to keep in mind is for any biological sample, you may have multiple transcripts being expressed in a cell at any given time at different levels. Maybe this one isn't expressed at all, maybe this one is a lot. So let's take a look at what our samples are telling us. Remember, this one was highly expressed in our statistical analysis, and this one was not very highly expressed. So even though they both have bumps, you could see in the scale that this one has a roughly 1,000 reads covering each of these exons by default, versus this one just having literally 1 or 2 reads. It's practically not expressed at all. Let's go ahead and hide this guy so we don't take up any more space than we have to. And as I'm going to just really dive into this guy, I'm going to make it a little bit bigger. And the other thing that we see in our pilot plot here is where the how many reads are spanning these introns. Remember, in transcription, you have the full length of the genomic region transcribed, and then you have a certain portion of that spliced out in a secondary step, and we're reading those spliced versions of those reads and then aligning them back, and we can see which introns have been spliced, but that also gives us an idea of which of these transcripts are being expressed in the sample. So you can see, for example, this first intron starting at this position, and you can see my mouse has got a dotted line which lines up exactly the exon stop, or where the exon stops and the intron starts, gets lined up with this sample. It's expressed at about 300 or 400 reads, and then that next transcript jumps it on up to about 600 reads. So both of these transcripts are being expressed in this sample. And then at some point, there is definitely more expression happening across this intron boundary, where all 4 transcripts have an intron boundary. So this is really interesting from a perspective of potentially doing information on each of these isoforms or versions of this transcription. You can also click on any of these notes or any of these features. You can click on any feature in GenomeBrowse. You can look at the actual transcript, pull it up in an external resource to understand if there's any information about it, etc. But now I'm going to start just actually exploring this a little bit more. I'm going to start zooming in on this exon. And as I zoom in, it is pulling in, literally tens of thousands of reads into a pile-up form. RNA-seq can have extremely high coverage. This can be quite challenging for some genome browsers to handle. But we are literally piling up tens of thousands of reads, and with average of 1,000 coverage here. And you can see actually there is a variant right in the middle of this non-coding region. Notice it's still in the UTR5 as I hover over it. The UTR5 for this in this gene, it's a non-coding region that might be a regulatory-oriented variant, so it might actually be interesting to look at to see if this variant is specific to all of the samples that have high expression in this gene. We could also see, is this variant very common inside public annotations? And to do that, we would add, for example, another annotation track. If we wanted to, I could add dbSNPs 135 common. If I add that, it would just take a second to load. But sure enough, that variant is actually annotated in dbSNPs. It's also annotated in 1,000 genomes, and I could pull those up as well. You can click on them, you can go to their RSID, you can link out to various resources that might tell you about that variant, whether it's associated with transcription, etc. So that just gives you a quick context of using GenomeBrowse to do RNA-seq expression profiling follow-up. It's very clean, it's very interactive, it's very visual, and you can definitely compare, not just one or two samples, and I have my screen at a low resolution here, so it's only so much I could display in a single view, but you can throw up dozens of samples in here. You can make these graphs tiny and just stack them all up and really go to town. So it's a lot of fun to follow up with your RNA-seq analysis. So let's go ahead and jump back into the mode of looking at where we are in our presentation. We've covered RNA-seq analysis, expression profiling. Now we're going to do DNA-seq putative variant validation. And what we're going to use for this is a really excellently done research project done by Dr. Golson-Lyons, like I mentioned, and a lot of his collaborators in Utah. And what they did was found a causal variant in the NAA10 gene. A little hard to read that, but you don't have to. I'll remember it. And what we're actually going to do is follow up on a couple different variant sites and end up in the NAA10 gene. All right, so we're using actually the samples themselves that, as I mentioned, Golson was gracious enough to share with us so that we could validate his results. We actually used SVS as an analysis and filtering platform to do the validation, and sure enough, we had no problem finding the causal variant as the only result of the filtering process using just modest filtering criteria. But what I'm going to do today is go through a couple different variant sites just so we get a feel for how this type of exome kit follow-up might look like. And this is not a whole exome analysis. As you can see, the coverage is extremely biased and in fact entirely centered around the X chromosome. I could double click on the X chromosome and center that. And now we have, as you can see, the affected individual here, and then no other individuals in the family were affected because it was a highly penetrant disease. We have a control uncle. We have the mother and the grandmother. It is an X chromosome disease, so as you can remember, this is going to only have a single copy of that chromosome from the uncle, from all the males in fact, from the affected child. The mother and the grandmother will have 2 copies, so their coverage over this region will be a little bit higher on average. And the unaffected brother, again, so less coverage because they have a single copy of that. And this is a X chromosome capture, so we will find that there is coverage centered around the exons of all of the genes in this X chromosome. And we can do, in our exploratory version of this analysis, fly into individual genes, hover over them, see them as they go. We can see where OMIM genes are. OMIM is a catalog called the Mendelian Inheritance in Man. And they catalog genes that have been associated with various diseases in a clinical setting. If you click on an individual gene up here, you can see what it's associated with, mental retardation in this case, in this gene. You can link out to it and read every single paper that was published that was associated with that gene. It can be a very useful tool for following up and doing your analysis. But we're going to do a version of, instead of just working on interpretation to start with, let's go ahead and look at an individual mutation and just see how well it holds water. So I'm going to copy and paste in here a variant, and again, the filtering process that we actually went through resulted in only that variant in NNA10, so I had to backtrack and find some interesting places to look at. But we are at the UTR3 region of this gene, and this region is obviously not covered to any great depth. It's got only about 10 reads at this position, but there is an insertion here of a GT, and if this came through the filtering process as being potentially interesting, this is a great example of showing how you might look at investigating, do you trust this variant? Do you send it to a lab to get Sanger sequenced and diagnosed officially as part of a patient profile, or is it potentially a false positive? The nice thing about GenomeBrowse is these are very easy to see. Even these insertions, which are essentially zero-width features, they exist in between bases. As I zoom into this region, I still have the context of where this insertion is. As I hover it over it, I see it's a GT insertion. I could see it's an insertion at the level of the reads, but already you can see that there are some of these bases that have very low quality. If I click on the individual reads, I get information about that read and the fact that the aligner didn't trust that this read was very well matched to this genomic location. The mapping quality is 0. That's usually an indication that this read could have just as well mapped to another part of the genome. So a little indication there that this does not look like a clean half, a hemizygous mutation as you would expect when you have a single chromosome. Let's go ahead and look at the other samples. Sure enough, it doesn't exist in the unaffected uncle, except for there is a single base that seems to have it. Now, maybe that's just by chance, but you don't get that kind of sequencing error usually. So there's something else going on here. That base is, maybe this is not a very clean region. The mother has it in a more clean homozygous state. You can see there is support for it on both strands. And similarly with the grandmother, actually has it in a heterozygous state. With a little bit of coverage, you can see it's about half and half, and the brother has it potentially in a homozygous state. So one thing I didn't quite show yet was being able to then, if I wanted to see, is this thing biased to one strand or another? Reads are always represented from which direction they were read from the genome. You would expect it to be on a strand agnostic sequencing protocol, about half and half. I can emphasize these mismatches so that they really pop out. I can also split by strand so that we can see the forward and reverse strand above and below the x-axis, and the fact that these variants are, do exist on both sides. So, what's going on here? Well, potentially with all those really bad mapping qualities, this is not a unique portion of the genome. You can read the actual nucleotide bases. It looks pretty repetitive. I have a track here called segmental duplications. What this does is just essentially flag any region of the genome that has repeat content. And sure enough, this looks like there's a part of the genome in chromosome Y that is very similar. How similar? I can click on it. I can scroll down. Looks like it's 100% similar on a base-by-base level, including insertions is 99% similar. So that explains sort of this crummy variant. Let's go ahead and jump to another region and look at that with GenomeBrowse. And the nice thing about this is it demonstrates, one, the display of a deletion, which is another interesting type of variant you expect to see at some frequency. And we might look at this level of this deletion and wonder, again, is it well supported? We can see here when we click on these reads that the mapping quality is quite high. The deletion seems to be nearly entirely covering by all reads. Not quite. We can zoom out to get a little more context, but there's not a lot of mismatches going on in this sample. The deletion seems to be represented in every single sample. So I would say these are two strong homozygous variants that are cleanly represented. And in fact, if we wanted to, it doesn't look like it's in the 1,000 Genomes database, but if I were to turn on, I have a couple other tracks hidden. If I were to show them, dbSNP does seem to have this variant. It lines up perfectly from its start and site. It's the GAT deletion, definitely another variant of interest. Or maybe, so now we're going to go ahead and jump to that gene that had the causal variant and spend a little time interpreting it. Now, it is in the OMIM catalog. It's an OMIM gene, and that usually indicates that there is some evidence that if it was to be knocked out of your sample, it could have clinical implications. It does not have segmental duplications, so that's nice to know. I'm going to hide that. There are variants here, but as we look down here on the bottom, and I've flagged this region just so I could see the whole genome context where this variant is, this right here in the affected child was the causal variant that fell out of the filtering. It's in a homozygous state. Let's go ahead and zoom in. Notice there is no variants covering this exon in 1,000 genomes. There is one variant in dbSNP. I could jump out to dbSNP, and I actually already did look at this. This does not look like it potentially is this might be a false positive variant because it was introduced in 2000, well, in dbSNP 129, not using modern sequencing technology, etc. But as I zoom into this, and you can see I have it flagged as with a mouse anchor. I'm going to clear that flag so we can look at it. It is very cleanly represented, so great representation of a clean variant. The individual reads look strong. They have high mapping quality. The read count is 100%. 96 count for the mismatch, 0 counts for the the match of the reference. The coverage for this exon is very strong, and as you go down to the other samples, it's not in the other males, and it's in a heterozygous state as you would expect in the carrier mother and grandmother. It is not in the other, in the brother. So what's going on at the gene level here? One, it looks like there are very few variants that actually cover the exons of this gene, right? The variants seem to be mostly intronic. So maybe this gene is highly conserved because it's extremely important to be functional. The other is the uncle, and in fact the uncle and the brother have a homozygous variant in this other exon. Now that variant, it's a hemizygous variant because they only have one copy of the X. So they've inherited this heterozygous mutation from one of the X chromosomes of the maternal line of this family, and the affected child inherited the other X chromosome. So there are two different haplotypes being inherited here. You can see that the mothers are the carriers for both, but when the child gets the unaffected, when the child inherits this haplotype, this chromosome, this hemizygous variant does not seem to be causal to any disease mutation, versus this one definitely was the causal variant. So it's very interesting to kind of confirm that there is a clean haplotypal structure that makes sense here. And finally, if we were to look at another track, the NHLBI 6500 exomes, this is a great track that represents over 6,500 individuals from the National Heart, Lung, and Blood Institute. And not only do they provide frequencies for each of those individuals, they also provide genotype counts. And if we were to zoom in to, say, a variant that does cover an exon, just to say, you know, is this gene potentially extremely important that it is not disturbed or knocked out? You click on this variant, and in the information console, we can really see that it occurs essentially only once in an African American individual, and never in a homozygous or hemizygous state, right? It only occurs as a heterozygote for the carriers. So that's extremely important, because that essentially says that that person probably is a carrier, and if this variant were to be inherited by that male, it might actually be, it might be just as damaging causal variant. So that's how we can validate and interpret those variants with GenomeBrowse from this excellent study and this excellent data set. So now for a little bit of fun, let's switch back on over and spend a little bit of time with my trios themselves. So what I have done is use some of the trio analysis workflows built into SVS. Preceding this webinar, we had our highest attended webinar of all time, actually, was done by Dr. Ken Kaufman, as he did an analysis of de novo variant analysis, compound heterozygous analysis, and rare recessive analysis of some actual clinical samples that he does in his lab. He used SVS as his workhorse, and we've actually even simplified the workflow that he's done since then. And he used GenomeBrowse to do his follow-up and visualization. So using SVS as a workhorse on my trio, I'm sorry to say, or really happy to say actually, I have, my son has no de novo variants of interest, no rare recessive variants of interest, and no compound heterozygous variants of interest. So what I had to do is back up and remove some of those common filters just to find some interesting things to follow up on here. And we'll probably just look at this de novo variant and one of these rare recessive variants, and then spend a little bit of time looking at one of my not so rare but potentially functionally interesting variants that 23andMe reported on my own exome. So I'm just going to switch projects here. Give me 1 second. And I'm going to drink of water. Okay. Excuse me. So we're loaded up at the whole genome context here. As you can see, I have those 3 exomes loaded, and unlike the X chromosome capture, these are well distributed across the entire genome. You can see the coverage is very even. It's coverage across all different samples. Here's the father, the mother, and my son. So here we go. Let's go ahead and start zooming in and using this information to interpret some of those variants. And I have in another buffer I'm going to copy over. This is a de novo variant that was discovered, but it really honestly was not a valid putative variant. I had to relax some of the filters and you'll see why. Because what we find here is, although it's in an OMIM gene, which would have been of interest, I'm going to hide that for now. And it is rare. It did not pass any of the, this got through all the filters of removing variants that were common in the population or even present in the population. It was called as homozygous for the father, or heterozygous, I'm sorry, yes, this is a de novo variant. It was actually called as, it was called as heterozygous, and homozygous, right? This is, and what we can see here is if I actually pull up the variant track itself, the variants for my son, you can see when I click on it the heterozygous genotype, and we can try to interpret what's going on here. It was actually called because there's called de novo because there's no reason why the son would be heterozygous while the parents are homozygous. It should have inherited the homozygous alternate allele. So why was the son called heterozygous? Well, for some reason, the variant caller decided that this was a heterozygous variant, and you can see that the ratio is not perfect, and that was one of the filters that actually removed this as a potential de novo variant. But it was called as heterozygous because it had 80 counts that matched the reference, 385 counts that didn't. Yet the parents were called as homozygous, but even they had some counts that did not match the reference. So if we were to try to interpret this, do I believe it? No, but what's really going on here, as I zoom out, you really get a sense that this gene is just sort of a mess, right? You've already seen a context of a really clean exome, a really clean gene, and this is what it looks like when you have a gene that is probably full of repeat regions that cover lots of different parts of the genome. And so you have reads mapping here that are, potentially aligned, but really have a lot of mismatches, and then you get this real spiky behavior in the coverage plot. So if I look at adding my segmental duplications track and showing that, sure enough, there is a segment duplication here that accounts for this guy. So not a de novo variant. Let's move on. So I also found a couple rare recessive variants, and these are essentially variants that are heterozygous in the parents and homozygous in the child. They are really rare. They're in a gene that's potentially interesting. In this case, the gene is in an OMIM gene. And the son, if I click on the variant track, is homozygous for this variant. So I'm going to go ahead and hide that for now. So here we are in the middle of this guy. And you can also see that the coverage is not consistent across. Yes, it's homozygous here, and if I zoom in, I can see the counts. And if I were to trust this, I might trust this a little bit more because it does have, fewer matches. But remember that homozygous variant we saw in Ogden? It was 100% matches. There's some other junk going on here, and at the whole exon level, you can really get a sense that there's a lot of mismatches going on. So I would not consider that a very interesting variant to follow up when, from a story perspective. So that's kind of just the trio level analysis, and let's go ahead and look at a variant that I covered in my blog post. It's an ERCC6. I'm just going to type in the gene name. It will search my tracks, give me a choice of what to jump to. And this variant covers is actually right here in this exon. You can see these little spikes here at the gene level. It's easy to notice where the variants are. But this variant was heterozygous in my case. But 23andMe reported it to me as one of the interesting variants of note in my genome. The reason is it's in an OMIM gene. It's relatively rare. If I pull up the NHLBI 6500s and 1000 genomes, it exists, and we will zoom into it to look at it, but it exists at a very low frequency, below 1%. It is heterozygous in my sample with extremely good coverage, and this is the kind of heterozygous variant you would trust. It's exactly half the number of reads. It's not present in the mother, and it's also heterozygous with very clean coverage in the son. So it looks like it was inherited from my chromosome to my son's. It's always fun to say that. Okay, so let's go ahead and look at this with a little more detail. I'm going to zoom in, and I'm going to zoom in on the base level just so we can see the context of these reads around it. And the interesting thing about this is it's right at the tail of this exon, right? So it's one base before that splice site. If it had been one base to the left, it might have really disturbed the splicing out of this intron and really affected this gene. This is an OMIM gene, and as I covered in my blog post, it was shown to be extremely vital and be be causal in a number of different syndromes if it was to be completely knocked out or to the level where it was extremely lowly, very low transcription of this gene. So let's go ahead and look at this frequency of this variant, see if it's of interest. And indeed, it's 1.9% among Europeans. So, if you were to follow a strict European level threshold, it would be considered potentially common. But it's at less than 1% of African Americans. And the interesting thing is also look at the genotype, right? So this is in chromosome 10. So you would expect to see either a heterozygous or homozygous genotype, and it occurs at a heterozygous state for a number of individuals, but not in a homozygous state for anybody. Nobody has both copies of those chromosomes containing this. That would probably indicate that, again, this is a very important gene, and it is haplosufficient in the sense that there are studies that show if you have a single mutated, the diseased version of this gene, you are just fine. And sure enough, I hope I am just fine. But if you were to have both copies of it knocked out, you would have some serious problematic syndromes. And again, looking at this from the genomic context, the reads are extremely clean. There are no segmental duplications. The presence of the gene in the variants catalogs is very well documented. So this would be, a potential carrier status level variant. I would not want any of those individuals who have it in a heterozygous state to meet up with any of those other ones and suffer the consequences of having offspring that would potentially inherit those two rare variants into a single individual. But in our case, it's no big deal. It is just a heterozygous, and it's not present in my spouse. So all good. Things are great in the world, but it's very fun to do this type of interpretation. I covered a couple of other variants in my blog, and you're welcome to go and view those. But this gives you a context of the level of interpretation you can really gain by using excuse me, by using GenomeBrowse and by using these number of annotation tracks. So with that, I am going to jump on over to wrapping this thing up, and we will spend some time answering some questions. So let's go ahead and jump back into slide mode. So as you get to the end of this, you will be given an opportunity. As I mentioned, we'll be sending out an email to you all with the download links for GenomeBrowse. I encourage you to jump on that and play with it today. And as I mentioned, we know we have not met all needs for all people. We have an exciting and aggressive roadmap for this product. It's going to be a free product. We intend to build on it in the future with all kinds of great value-added solutions as well. But please use it now. It will always be free. But also, what we're going to do is launch a support site and questions and feature request site for this for GenomeBrowse. You'll be able to use your login to log into that, your GenomeBrowse registration. You'll be able to ask your questions, and we really hope to encourage a lot of community discussion about best practices or tips and tricks and ways in which you can use GenomeBrowse to accomplish your needs. But also, as I mentioned, Golden Helix is a very interactive and stakeholder and feedback-oriented company, and we want to hear from you guys. How are you using GenomeBrowse? How would you like to use it? What are the most important things you could imagine that would help you and make your life easier? And send us those feature requests through our community support site. Until we have that launch, though, what we're going to do is open up GenomeBrowse at goldenhelix.com to be a proxy for that. It will be a line to myself and our fabulous bioinformatics support team. And we would ask that you be open to making all of your conversations on there feeder questions for our support site, for our community site, so that when we do launch it, we'll have some good content already up and going so we can start having those great discussions. So Thank you again for spending your time here with this great product launch. And I know you probably already have your iPhone launch rumor site feeding as it goes, but let's go ahead and spend a couple more minutes going through the questions that we have coming in. So I have a number of questions coming in, and man, it's going to take me a second just to plow through these and see which ones we want to answer. But thank you again for your attendance, and stick on the line and we'll answer some questions, and then we're on our way. Okay, so I'm just going to go ahead and plow through these as quickly as, or in the order if I can. One question is just, what is the address of our blog? It's easy to find by going to goldenhelix.com. We promote it there, but it's blog.goldenhelix.com. There's also a section on that blog called Gabe Informatics. That's where I blog directly, and you can subscribe to the blog in general, or you could subscribe to my feed as I do my work in building the annotations and interpreting genomes and being part of the product team. So please feel free to access that blog. It's a great way for us to communicate what we're thinking and for you to be up to speed on how things are going. And another question about whether this webinar will be available. Yes, it will be. I mentioned that in the beginning, but for those who didn't hear that, we will record it and we will make it available on our website, and hopefully get a lot of the ums out of my speech, and we'll just make that part of the standard Genome Browser online help directory. So next question is, do you have do I have standard visualizations for the type of statistical analysis that you would expect inside of SNP and Variation Suite? And so that's a very good question. As I mentioned, our SNP and Variation Suite already has a visualization tool that incorporates all of these same public annotations, and that's what we're using right now inside of SVS. The intention is, and the plan is, to move GenomeBrowse into that framework to be the visualization tool for all of those statistical results. And also in our roadmap, we intend to be able to take your own statistical or other types of genomic features and bring those in as tracks. If you're an SVS customer, that is already a possibility, because SVS can convert any data source to a genomic annotation track visible by GenomeBrowse, but that is on our roadmap as well to make that a standalone feature. So yes, stay tuned. You'll be viewing beautiful Manhattan plots GenomeBrowse style in a matter of months. All right, can I get, thanks, good. You're welcome, you're welcome, you're welcome. And what is the y-axis on the reference sequence gene? Could it be read depth? So that's a good question, and actually might as well be in GenomeBrowse mode to answer that. So you can see we actually don't have a label here. The y-axis for some of these graphs that are essentially just genomic features that potentially overlap, whether it's genes, whether it's OMIM itself. I'm going to go ahead with OMIM, zoom on out to the level where you can see that there's a stack here. That's just the stacking of those features. There happens to be 6 different OMIM entries or 5 different OMIM entries that have the same start and stop site. That's not statistical, or that's not significant from a depth perspective. The y-axis is simply just the amount of features that lap there. The difference is always when we are looking at something like the reads. Those reads imply a depth when you stack them up, and that depth is significant from an interpretation perspective. Are there animal annotations? That's a really good question. We actually have, as you can see in my little drop down of different genome builds that are pre-built into the software, and they're easy to add. We have a number of animals, lots of mammalian species, different cattle builds, dog, and actually this list goes on and on and on. As I mentioned, Golden Helix has been doing this type of work on building public annotations and putting them into a visual context for a number of years, and we can actually piggyback on all of that existing support and technology and curated annotations. And for a lot of these species, there will be reference sequences, there will be genes, there will be a lot of other annotations available. All right. Did you try GenomeBrowse on FFPE extracted nucleic acids? That's a really good question, and the ultimate answer is it's going to come down to a lot more important decisions than which genome browser you use to get the quality out of a formalin-fixed wax type sample. You're going to have the right library preparation before putting that sample on a sequencer. The sequencing protocol might need to go to an extra read depth to account for the natural degradation of those DNA material. And then potentially your alignment and filtering tools need to crop out all of those bad reads. The type of view you have here with just this gorgeous pile up with very clean variants, and you can call these. You could say heterozygote, right? Reference. That comes from having an upstream secondary analysis pipeline that has already filtered out reads with low quality or trimmed reads that have gone off kilter in the sequencing process. And so there is a lot of work upstream to produce an interpretable result that gives you this easy of an interpretation job. Can I load my old BAM files? I hope by old he means just using potentially a different genome build. Yes, we support HG18. There's a number of annotations from it. You can actually open up any BAM file just by going to File. You can create. I have a number of different BAM files from the Ogden syndrome, from my own trios. Basically, we do aspire to that goal, as I laid out in the beginning, of making this as low friction as possible to get your data in. If your BAM file is not indexed, if it doesn't have the coverage information to display this forward and reverse coverage at these large zoom levels, we'll do that for you automatically in the background while you work. A new file will just say, I'm computing coverage, I'm building that index, and then when it's done, boom, it loads right up. So yes, if you have BAM files lying around, as we all do, sitting on those giant hard drives from when we first got our data, jump in, add that hard drive as a source, and start looking around. With the RNA-seq analysis, can you visualize the amino acid sequence of the gene's protein amino acids correctly enumerated? That's a really good question, and that is essentially a feature request because we cannot currently show those amino acids that occur every 3 bases of the coding sequence of a gene. That is something we have in our queue, and we plan to support that so you can see each amino acid letter as it is, but that also implies that you take account of variation to see when an amino acid changes from the reference. So that's a very good question, and please put that as a feature request to us, and I'm sure we actually already have that, so we'll mark that as a upvote on that feature request. Your exome chromosome example is in the PAR region, which is identical in X and Y. So I think I jumped around a lot, but the region that we were looking at in that final gene definitely was a unique region of the genome, and hence we could see those very clean reads, and that variant was validated. Can you clarify on the login accounts requirements? When information is going to be tracked? That's a very valid question. So basically, yes, we do require you to create an account and log in. We mostly do that from a support perspective and from a perspective of being able for you to have an identity in the community. We want you to be able to log into our support site and be able to set a profile picture and share your best practices and engage in conversations there. We also are making this available for free so that you can access all these public annotations. But we do want you to be able to know about any of our upcoming events. We do have a way of you saying keep informed as you log in. If you want to turn that off, we won't send you any notifications of our upcoming events. But we aren't, going to be doing much more than just making sure that as you log in, you have an account, and that in the future may include access. And currently does include that account logs you into your EA pipeline account. So we have a cloud-based annotation server for RNA-seq samples. If you're an EA customer, your account is automatically linked up there. And we want to be able to keep that open for doing things like base-based integration in the future. So your account is essentially your way to access all these great resources. Can read quality contained in the data console be used to filter the data, or could something like the average quality be extracted and loaded as a track? So that is a great question, because it really gets into, what is the goal of this tool, and what are sort of the bounds of that analysis goal. And so ultimately, this is not a full-blown analysis suite. We don't intend to put in here every feature that would allow you to filter on read depth, or essentially analyze these large files. We find that some of the BAM analysis does make sense to do in pipelines like these secondary analysis pipelines, because they are extremely computationally intensive, and the filtering analysis makes sense to do in a tool designed with that type of workflow in mind. SNP and Variation Suite has been built out to handle both RNA-seq and DNA-seq variant analysis, rare and population level analysis. And so this is a tool to kind of take those filters and visualize them. So it is a visualization-oriented tool. That said, what we were planning on doing in the future is soliciting again from the community the interest in a tool to do more integrative filtering from this interactive interface, and you'll be hearing more about that as that progresses. What species are loaded? I already covered that, so thank you very much. We do have a number of species loaded. Can we use this for bacteria genomes? So, ultimately, a genomic reference is required, but a lot of bacterial genomes do have very good references. So if you have a genomic reference, you have a reference sequence, you have a genome build, and you've aligned to that reference, this is a great tool for that. If you're doing a profile of many different subtypes of species, or you're doing a lot of de novo assembly, you can definitely use this to view your de novo contigs, but it is not going to necessarily benefit from having all that information that can be publicly curated based on a reference. So I hope that gives you a little context about what we're focusing on there. Can you overlay a track of mapability? Mapability is another great annotation track like segmental duplication to help you give an idea of how good an individual region is at being unique and being able to be well assayed by the short read technology of next generation sequencing. Our reads are in this case, we click on a read, we can see how long it is. We have reads that are on the order of 76 bases in this case. So that type of read length will find difficulty in those regions of the genome that are highly repetitive, and we could use a mapability track to help us determine those, just like segmental duplications. I don't think we have we don't have mapability as a pre-curated track. I know it's on my data curation roadmap, but again, with SVS you can load in those types of tracks from one of those repositories like UCSC and create a track for that. Let's see. Let's just go to another couple more minutes, and I certainly am discovering there's a number of questions here that I'm not going to be able to get to. I have probably 40 questions, and at this pace I won't get to them. But please continue to ask your questions, and what we'll do is we'll try to follow up with you. We have your name that you use to log into GoToMeeting, and we have and you can certainly feel free to email GenomeBrowse@goldenhelix.com. Excuse me. Do you have the ability to analyze chip-based expression analyses such as Affymetrix cell files? So obviously, if you're using a platform like Affymetrix to do your expression profiling, you do have some probe level information about those intensities. You don't have the full RNA-seq glory like we were using earlier, but that information can be pulled in as a track. It gets into the realm of sort of looking at statistical or probe level information as opposed to sequencing data, so that is not, since we don't have all these custom track building tools built right in, you would have to use SVS to build that track, but you could certainly imagine, especially in the context of comparing an expression array to an RNA-seq experiment, using both in the same visual context. Can you use GenomeBrowse for feature creation, and how should it be cited? An excellent question. I'm glad I got to that one. Right now, what we're suggesting is that you take a screenshot of this beautiful interface. You can do a lot to customize the essentially the level of details, the fonts. You can open up the font editor. You can change the titles to look as big or as small as you like. You can move anything around. You can really make this publication quality. We really spent a lot of time on that because we did find that was something people were asking us about and seeing another Genome browsers falling short on. The other thing that we do have in our roadmap is to make a screenshot form of this for you automatically to essentially create a PNG of all of your graphs in a way that removes some of the user interface elements. But right now, as is pretty common actually in the Genome browser community, feel free to screenshot and Photoshop this thing, and please cite us. We would just cite us through a web reference to Golden Helix, and we plan to publish a paper about sort of the unique things that we were able to accomplish in our journey to build this thing. But in the meantime, please just cite Golden Helix's website and the Golden Helix software for GenomeBrowse. Okay, so I'm 10 minutes past the hour, and I would love to continue on this conversation, but we all don't want, it would probably take me another half an hour, honestly, to plow through these questions. So let's take this offline, as they say, and move our questions to email form, and then as we launch our community site, we'll be doing this type of interaction there. So thank you again, everybody, for your attendance today and making this a great product launch. I'm very excited to see how this is going to be used, because every time a new person touches a piece of software that we've built, we find that they have some meaningful and unique way in which they use it, and it just keeps us going at night and all these long days and hours we put on it, knowing that it's going to be used for those great purposes. So with that, go forth and download GenomeBrowse and start engaging with the community. Thank you very much.